What time is in your country, Rebaz?
It's 9 p.m.
Oh, wow.
Yeah.
They already had dinner and everything.
Yeah, I have a few family members actually from Europe. My uncle is trying to figure out the Zoom, I guess.
I see.
38 participants.
Hey, buddy.
Hey, Carly. Thank you.
I texted you a bunch of times because I was having issues getting in, but I obviously found my way.
Okay, perfect.
You look great. Good luck, buddy.
Thank you, appreciate.
Can you see me, Rebaz?
Tell them if they have questions they can ask. Mama, Can جلما؟
عامل جملته.
دست
توم
خوش.
نبینید؟
بله.
دست خوش. باش زور باش. زور باش. هدیه جايبینه ان شاء الله.
باشه با قربان بخیر بنی اخوا.
سپاس وش سکوتو بیت.
يا اخوا بيويبي.
ماما شلون يحفظ دنگو باسه.
كاكا زانا جان زور سپاس ممنونم.
ان شاء الله پیروز بی و سرکوتو بیت و ان شاء الله نخشه به لیمش.
ان شاء الله برام دست خوش به زور سپاس بو هاتنت. Dr. Jan?
Jan.
Did you figure out finally?
Yep.
Okay, great. Sorry, I have been like really busy. I didn't get a chance to like, email and all of that.
No problem. My bad, actually. I figured out you sent me that picture before, but I just didn't notice. So no worries. You're okay.
Assalamu alaikum, brother.
Wa alaikum assalam. How are you, brother?
Alhamdulillah, alhamdulillah. I'm going to stay muted and off the camera, but I just, I'm really proud of you and may Allah make you successful. And, I'm really happy for this day, man.
Thank you so much. That means
I'll be here for you, inshallah. Allah ma'akum.
Rose, I have a lot of family members that they don't speak English, so I have to communicate with them.
That's not a problem. Great.
Everybody else has their cameras on. Camera.
Wow, I haven't seen in a long time a thesis defense, this populated. So you might have a lot of friends, I'm sure.
Yeah, Dr. Maria, I have a lot of friends.
You too, yeah.
And actually, my defense was announced in Kurdistan as a news, so everybody had it. They had the information.
That's great.
More pressure on me.
Rebaz, are you ready? It's almost time.
Yes, I am ready.
Brittany Chicken says hello. Hyder.
Okay. I can see Lane there. Hi, Lane.
Yeah,
Ramesh, it's good to see you.
Yeah, nice seeing you.
It's been a while.
Yeah. You moved to a different building, so we don't see you anymore.
Yeah.
Hey, Lalo.
ریباز گنچونی سرکوتو بی انشالله.
زور سپاس آموزین جان. ممنونم دست ماجکم.
خوش بی.
Okay, so let's start 12 on the dot. So let me first introduce, hold on, Rebaz, hold on. Let me introduce Mr. Rebaz Ahmed and give a little bit of introduction. First of all, good afternoon to all in the United States, and good morning or good evening for those who are participating from outside the United States. As you all know, it's a big day for Mr. Rebaz Ahmed, the first time I'm mentioning his middle name, as he is ready to defend his PhD thesis today and graduate to receive the highest degree, namely the PhD. It will be a day for him to remember in his life, as his dream to receive his PhD will become a reality. In a couple of hours, his reference will change from Mr. Rebaz Ahmed to Dr. Rebaz Ahmed. I'm sure Mr. Rebaz Ahmed's family is very proud of his accomplishments, and to witness him successfully defend his thesis and graduate. Though they are far apart in Kurdistan, their wishes and support are here with him and can be felt. And again, thank Mr. Abbas's family for participating to see your son graduate or defend his thesis. Just to give a little introduction about Mr. Rebaz Ahmed, that he joined my lab as a PhD student sometime in April of 2016. I still remember he walked into my office with great confidence, and he told me he has the experience and has mastered a lot of techniques during his master's degree way back in India. So my first reaction, which is normal, is great. You seem to exude with confidence, and I said, well, I appreciate your confidence, but my first step is you will need to demonstrate that you can do what you claim in my lab. So pretty soon he realized there is a lot more to learn in research, and research is not easy. So while he struggled in the first year, I can say with confidence that he did struggle in the first year. He felt that I hope he did not feel that I was harsh on him. Even today, I sometimes feel maybe he feels that I'm harsh on him. But the training on PhD is not to become a super smart guy in 2 days. It's an experience. You learn how to master the techniques and how to do different things in life. And that takes you throughout your life. So in the 4 years, he has quickly mastered the techniques, and his research has moved pretty rapidly. And today he's ready to defend his thesis. I find him to be a very bright and smart and easygoing guy, always being very helpful in the lab and very cooperative. And I strongly believe he's ready to take the next step in his career and move forward. I wish him the best in his future endeavors and look forward to seeing him make big breakthrough in science and make a big difference in cancer research, any other field that he chooses going forward. Finally, but not last the least, I would like to thank all the committee members, Dr. Zhou Zhao, Dr. Wei Ching Ding, Dr. Jerry Wu, Dr. Courtney Griffin, and Dr. Maria Ruiz Echeverria for their support and guidance provided to Mr. Rebaz throughout this 4 years of his PhD thesis. So just to give a little bit of the format, so I'm going to hand over the mic to Rebaz, and he will present his work and defend his thesis. So during his presentation, I request all the participating friends and family and the committee members to mute so that there's no additional disturbance. Sometimes the Zoom can get interrupted with audio and video. And after his 1 hour presentation, I would request the committee members to first go with their questions. And later on, if the audience, anybody participating have any questions, you can actually type in the chat box. Then I would ask those questions in the order it is received for Mr. Rebaz to respond. And after the 15 or 20 minutes of his defense completion and questions and answers, I would request all the non-participating committee members to turn off their video, and Rose will take care of it. And then the committee members will discuss with me in the, and I guess we will ask Rebaz to step out and then step back. And that will be the format for today. And once again, I appreciate everybody to participate in this presentation, and I look forward for Rebaz's presentation. Rebaz, it's all yours now.
Thank you so much, Dr. Ramesh, for the wonderful introduction. And good afternoon, everybody from the United States, and good evening for those who took time to pick into my defense, defending my thesis. As you all know, I came from a beautiful city, which is the cultural capital of Kurdistan. Here, I just wanted to show a few pictures of my city, and also I wanted to show you some of my travels to seek science and getting knowledge all over the world. But the two scholarships that I got, one of them from Germany and the other one as a Fulbright scholar from the University of Central Oklahoma, actually took my knowledge to the next step, and I wanted to pursue my PhD. And for that, I accepted the PhD offer from the OU Health Science Center, and I joined Dr. Ramesh's lab to study the most common type of lung cancer, non-small cell lung cancer. And actually, lung cancer is leading cause of cancer-related mortality in the world, in both male and female. The American Cancer Society estimated over 200,000 people will be diagnosed with this disease, and more than half of them die from the disease. And I lost my hero in 2017 from lung cancer. So as you can see, lung cancer is very personal to me. The low survival rate of both types of lung cancer is actually because
Rebaz, continue. I think Ross might be able to control the mute.
Okay. So the low survival rate of both type of lung cancer is mainly because of the diagnosis. Because when the patients are diagnosed, they are already in the metastatic stage. That's why the conventional therapy do not work for them. But thank technology that enabled us to identify the molecular driver of cancer initiation and progression, and also develop new therapies.
Rebaz, can you put your slides in a presentation mode? I think it's too small for people to see it. Can you make it like a single view? Yeah, there we go. I guess. Can you do it in presentation mode? Can everybody see it? Better?
Yes.
Is it better now?
No, I think maybe you might want to go to the previous one then.
Yeah, Rebaz, we're getting your kind of the presenter view instead of just a single slide.
Yeah, maybe this might be better.
Okay,
No, he can just go to the display settings and change it. If you want to go to the presentation, go to the presentation first. And on the top, you have display settings. Change it to presenter mode. Swap presenter, that's what. Click that. Yeah.
Is that better?
Perfect.
All right, thank you so much. This is called technical issues. Okay. Anyways, the problem with these patients are when they are coming to the clinic to be diagnosed, they are already in the metastatic stage. That's why the conventional therapy do not work for them. But thank technology that enabled us to identify the molecular driver of cancer initiation and progression so that we could go after the cancer cells and spare the normal cells. And one such molecular driver is called epidermal growth factor receptor, EGFR. As we can see, EGFR is overexpressed in many types of cancer, including lung cancer. But why overexpression of EGFR is important for the cancer cells to survive? Because normally when EGFR, and when I do that, my stuff doesn't work. Okay, so EGFR, when the ligand binds to EGFR, it causes dimerization and activation of the downstream signaling pathways to support proliferation, cell growth, and cell survival. That's why it's not surprising that the overexpression of of EGFR on the surface of the cancer cells will lead to increase the activation of these signaling pathways that can further support the cancer cells to survive. That's why EGFR has been a molecular target for cancer therapy for more than a decade. And in fact, we have, up to date, we have 3 potent generation of EGFR tyrosine kinase inhibitors in clinic that only benefit the cancer patients that they have activating mutations in their kinase domain. Although after 1.5 years into the treatment, they get resistance. But on the other hand, we have a good number of patients that they initially either respond or not respond. There is a contradicting ideas about their response to all of these generation of EGFR inhibitors. So what could be the mechanism? And here I am, started working, joining Dr. Ramesh's lab and reading about the importance of the chromatin remodeling complexes in cancer formation and treatment response. And one of them is the most evolutionary conserved complex. Is called the Swiss-Nef complex, which is a multi-subunit complex. It has got 2 important ATPase core subunits that they are very important in a functional of the complex. But these are not independent. They get signals to help with gene expression from the cytoplasm or from outside of the cells. And they could be getting signals from EGFR. So this is where I have given during my PhD time when I started, I have been given 2 flasks of cells and I immediately grow them, I seeded them, and I treated them with the clinically relevant concentration of gefitinib. And gefitinib is a potent EGFR inhibitor and it's FDA approved. As we can see in both the cell lines, first of all, I picked the cell lines very carefully based on their mutation status. Both they got wild type EGFR, but they have different mutation of their BRG1. And gefitinib actually was able to downregulate the phospho-EGFR in both the cell lines. But what was the great observation was, in the wild type BRG1 cells, we have downregulation of EGFR, but the total EGFR in the mutant BRG1 cells did not change. That's number 1. Number 2, I would like to draw your attention to the expression of BRG1 and BRM, both the core subunits of the Swiss-Nef complex, that gefitinib actually increased the expression of both mutant and wild type BRG1, but no change in the BRM expression, indicating that there could be a link between EGFR and BRG1. And what is BRG1 now? There is a lot known about BRG1, but I just highlighted a few important points. That's very important in tissue maintenance, mammalian development, and stem cell homeostasis. And also recently, the role of BRG1 has been shown in cancer formation, as well as response to anti-cancer therapies. And BRG1 actually is known to be a tumor suppressor in lung cancer because its mutation, non-functional mutation, has been reported in almost 35% of the non-small cell lung cancer cells that we use them for research. And also almost 10% of patients, they have loss of BRG1. But what is interesting is BRG1 actually is promoting survival, more acting like an oncogene in other type of cancers such as neuroblastoma and melanoma. And at the end of my presentation, we will talk more about this. But then we want to know what is the relation between EGFR and BRG1. I have done a lot of experiments, but here I'm showing you the key experiments that hopefully can convince you what is the relation between BRG1 and EGFR. First, with a specific siRNA of BRG1, we downregulated BRG1 indicated by the downregulation of both mRNA and protein level. And the EGFR expression at the protein and mRNA level shoots up. But interestingly, although we have downregulation of BRG1 in the mutant BRG1 cells, but no change was evident at the mRNA and the protein level of EGFR, indicating mutation of BRG1 could be very important in the expression of EGFR. Then I overexpress BRG1, this is silencing. Then this time I overexpress BRG1 in these two cells to see what happened with EGFR. We can see overexpression of BRG1 both at the mRNA and protein level actually downregulates EGFR. And in a separate experiment, this time I used a specific EGFR siRNA to silence EGFR and see what happened with BRG1. We can see the siBRG1 significantly reduced the mRNA and the protein level of EGFR in both the cell lines. But although we have upregulation of increase in the mRNA of BRG1, we did not see significant increase in the BRG1 expression. And the gefitinib treatment, I just showed you some data that the gefitinib treatment actually reduces the phospho-EGFR in both the cell lines, and it upregulates BRG1. But again, what was interesting to observe here is the total EGFR. It changed, it reduces when we have wild type BRG1, but it does not change when we have mutant BRG1, suggesting a negative relation between BRG1 and EGFR, and the mutation of BRG1 is very important for EGFR to be expressed. But this is all in culture dish. To be more clinically relevant, we got tissue microarray from patients with the healthy control tissues, stained for both EGFR and BRG1. And we stained these tissues using a PIRIO system. First of all, we noticed that the expression of BRG1 and EGFR both are significantly more compared to the healthy tissues. And we further dissected this result, and we found that we noticed that 75% of the tissues had high EGFR and low BRG1, and the 25% of the tissues had low EGFR, high BRG1. Again, indicating the negative relation between BRG1 and EGFR. What does it mean, this negative relation? It means that these two proteins are in perfect balance to support tumorigenesis. But clinically, when we shut down EGFR, it's up to the mutation of BRG1 to determine the efficacy of EGFR to treat non-small cell lung cancer patients or the cell lines. To test our hypothesis, we found 3 specific things. Let's go one by one and the supporting data that we gathered to further prove our hypothesis. First, we wanted to see the role, we wanted to test the role of BRG1 in the resistance of non-small cell lung cancer to EGFR TKIs, and we used gefitinib as it's FDA approved and it's very potent, widely used in the clinic. I treated 3 different cell lines, again with the clinically relevant concentration of gefitinib at both 24 and 48 hours. We can look at the wild type EGFR cells first. When we have wild type BRG1, they respond better compared to the mutant BRG1 cells that they exhibited resistance at all the concentrations tested. And we have used HCC827 as it's known as super sensitive because it has got the activating mutation as the control. Then the molecular studies from this experiment showed that all the tested markers downstream of EGFR, they were reduced in the super sensitive cells. But in the wild type EGFR cells, based on the mutation of BRG1, they responded differently. Here, I just want to draw your attention to the expression of EGFR and phospho-AKT because we will need them later. I will remind you about this. Phospho-AKT in both the EGFR wild type cells were upregulated upon gefitinib treatment, but the total EGFR was not affected, while the total EGFR in the wild type BRG1 cells was reduced. We also noticed that the BRG1 actually was reduced upon gefitinib treatment, but not in the wild-type EGFR cells. So we wanted to silence BRG1 in these cells and followed by gefitinib treatment to see what happened. And here is the result. Interestingly, the A549 mutant BRG1 cells, when we silence BRG1, they don't care. They don't show any response. Neither the expression of activated or total EGFR. And when it was followed by the gefitinib treatment, in the combination treatment, again, we don't see enhanced activity of gefitinib. But on the other hand, we see the wild-type EGFR cells, when you take away in the wild-type EGFR and wild-type BRG1 cells, when you take away BRG1, actually they are not surviving. It significantly reduced their cell viability, meaning these cells are somehow dependent on BRG1. And when you followed it by gefitinib, further enhance the inhibitory effect of gefitinib treatment, which can be explained by the reduction of all these markers tested upon the dual treatment. This is very interesting. Then we modulated BRG1. We created stably knockout and stably overexpressing cell lines in our lab. First, let's go with the overexpression. This is the cell line that the A549 that has mutant BRG1, they have minimal response. They exhibit minimal response to gefitinib treatment. But when we put back BRG1 in these cells, we see the cells started responding significantly to gefitinib. One thing also we need to note that here, the BRG1 overexpression significantly increased the cell viability of the A549 cells. And this sensitivity can be explained by the reduction of all these markers when now they have overexpressed BRG1 compared to the control cells. I just showed you one slide before that when the cells are having wild-type BRG1, they will be sensitive to SI-BRG1. Then we wanted to apply the same concept in these cells, and we targeted BRG1, silence BRG1 with the SI-BRG1. We can see the cells, those cells that they had mutant BRG1 did not respond to SI-BRG1. Since now they have wild-type BRG1, they significantly started responding to taking away BRG1 or silencing BRG1. And when it was followed by gefitinib treatment, it was further enhanced, the activity of gefitinib treatment. Again, which can be explained by the downregulation of all of these markers except for total EGFR upon the dual treatment. Now let's move on to the knockout cells. We created single cell clones using CRISPR-Cas9 technology to knock out BRG1. We can see the control cells that they have wild-type BRG1 again. They kind of dose dependently reducing to all of these concentration of gefitinib. But when you knock out BRG1, when you mutate BRG1, they do not respond. And also, I had to increase the concentration of gefitinib to almost 20 micromolar to get roughly about what I get at 1 micromolar in the control cells. Because the control cells have downregulation of both phospho, total EGFR, while minimal of phospho-AKT upregulation was noticed. But in the knockout cells, the phospho-EGFR started maybe reducing at 10 micromolar, and that's what we see in the cell viability, or at 20 micromolar that they started responding. But what is interesting here again, the expression of EGFR and phospho-AKT, that they actually move parallel. They parallelly increase upon the gefitinib as we increase the concentration of gefitinib. This is another indication that I'm telling you, we need these two proteins later to find the mechanism. Moreover, taking away BRG1 from these cells dramatically changed the morphology of the cells. Therefore, we put them on the migration and invasion chambers to see if these cells are aggressive. And the answer is yes. We can see here the minimal cells where the negligible number of control cells were able to invade. However, we have significant number of, or tremendous number of knockout cells were able to pass through the migration and invasion chambers. And more interestingly, gefitinib treatment did not mitigate the aggressiveness of the knockout cells. And now if I show you this data, I know you will be really eager to see if I put back BRG1 in these cells, in the BRG1 knockout cells, and see what happens. And here is the result. The knockout cells that they did not show any response based on the cell viability to 2 micromolar gefitinib after putting back BRG1 with the help of PMX BRG1 vector, now significantly started responding to gefitinib treatment. Again, which can be explained by the downregulation of EGFR and phospho-AKT and the other markers tested too. And this data is very exciting. I still remember the excitement of all the lab members that I walk into Dr. Ramesh's office, and I told him, Dr. Ramesh, it's been 7 days, we transfected the cells with PMX, and we see colonies of cells that they restored their morphology more like the control cells. And we discussed why this AKT is important, the phospho-AKT, because we see the resistance of the knockout cells is mostly due to the upregulation of EGFR and phospho-AKT. And we wanted to target AKT this time and see if these cells that they are not responding to gefitinib, but they are responding to AKT inhibitors. And the results showed that, yes, the knockout cells that, again, did not respond to 10 micromolar gefitinib, now they are responding to minimal concentration of AKT inhibitor, the MK2206. But to our surprise, that the combination treatment actually did not enhance the efficacy of gefitinib. Now I have all the experiments set based on gefitinib treatment, which is a first generation of TKI. We wanted to know whether the role of BRG1 is limited to the first generation or is for all the EGFR TKIs. To do that, I treated the cells with the osimertinib, which is the third generation, the most recent potent EGFR inhibitor. And interestingly, we see that the cells, the A549 mutant cells, exhibited minimal reduction in cell viability, while at the same concentration tested significantly reduced the cell viability of the BRG1 wild type cells. And the molecular studies showed that, again, we have the upregulation of phospho-AKT, but in the sensitive cells, the wild type BRG1 cells that they are sensitive, we have reduction in total EGFR, but not in the mutant A549 cells. This is telling us that, yes, BRG1 is very important. Its mutation is very important in the response to EGFR TKIs in general. Second is telling us that EGFR mutation screening could be not the only parameter that we have to pay attention to give treatment to the patients. Moreover, if we have mutant patient BRG1, they could be getting benefit from the AKT inhibitors, not the gefitinib. Now I have solid data from the in vitro study that gave us enough confidence to move our study to the in vivo study. We implanted the control cells with the wild type BRG1 in the flank of nude mice and let them grow, and then started the gefitinib treatment and monitored them for almost more than 22 days. We can see gefitinib treatment significantly delayed the tumor growth in the gefitinib treatment compared to the vehicle-treated cells, gefitinib-treated mice. Again, the molecular study showed that in these wild-type BRG1 cells, upon gefitinib treatment, they will have reduction of EGFR. Then we have the mutant BRG1 cells that we implanted again in the flank of nude mice. We can see these tumors do not care about gefitinib treatment. The graph is actually moving in parallel throughout the study. But the same cells, when we put back wild-type BRG1, the tumors actually significantly, the wild-type BRG1 tumors significantly delayed in their growth compared to the vehicle treatment. And again, it's because of the EGFR and phospho-AKT overexpression in the mutant BRG1 tumors. But when we put back BRG1, we can clearly see the downregulation of these two markers. Then we collected the tumors and stained them for H&E and Ki67. And the quantification of the Ki67 also showed that when in the wild type BRG1 followed by gefitinib treatment, significantly reduced compared to the other groups. And I had more slides. I stained them for the other markers. And then again, quantified them using the Aperio system. That again, BRG1 is increasing, phospho-EGFR is reducing. But what I wanted to point out here again is the expression of EGFR and phospho-AKT. In the mutant tumors, when you treat them with gefitinib, you have increase of phospho-AKT and EGFR compared to the controls. But then when we have BRG1 wild type, followed by gefitinib treatment, it significantly reduced the intensity or the expression of these markers in the tumors. Again, further supporting that there could be an interaction between EGFR and phospho-AKT governed by BRG1. Now we are confident that BRG1 is playing an important role in treatment response, both in vivo and in vitro. And now my favorite part is going to start, because I like to dig into these pathways and see how BRG1 is controlling this resistance. Accumulated evidence from the literature showed that either EGFR by itself or AKT or the ratio of the expression of both are very important in treatment response or treatment outcome. And in another study, they showed that when EGFR is internalized, it binds to AKT and AKT shuttles it to the nucleus to help with all genes that they are supporting the proliferation or metastasis of the cancer cells. And I would like to remind you that in my knockout system, I saw the upregulation of both EGFR and phospho-AKT upon the gefitinib treatment. So the question here is there any physical interaction between these two? But before doing that, I wanted to further confirm, because we are peaking. I wanted to confirm that when we have knockout BRG1 cells, they have more expression of EGFR in all the cellular fractions compared to the control cells. And just for EGFR, I further proved it by the flow cytometry to see that, yes, EGFR is, the expression of EGFR is more compared to the control cells and phospho-EGFR. And now I pulled down, now I'm confident about this. So I pulled down first EGFR and detected the associated EGFR with it. And in the reverse experiment, this time I pulled down phospho-AKT and detected the associated EGFR with it, suggesting the EGFR and phospho-AKT is actually binding to each other. Does gefitinib treatment disrupts this interaction? And the answer is no, because in both the knockout cells, I see that compared to the DMSO treated cells, the interaction between these two, I was able to co-IP them, so the interaction was not disrupted. Neither in the A549 cells that they both have wild mutant BRG1 cells. So to disrupt this interaction and make the cancer cells sensitive, we have to treat them with AKT inhibitors. Because when we treat the cells with AKT inhibitors, of course, the pull down phospho-AKT will be less, and then the associated EGFR was less too compared to the DMSO treated cells. And actually in the A549 cells, when there was no AKT, almost all the AKT, phospho-AKT was eliminated, no EGFR was detected. Again, further suggesting first the interaction between phospho-AKT and EGFR holds true. Second, BRG1 mutation is governing this interaction to happen. Again, this is in vivo. It's all in culture dish. But I had my slides, IHC slides, stained for all of these markers, but now I wanted to focus on EGFR and phospho-AKT. And Aperio system allows us to quantify the membrane, cytoplasmic, and nuclear fraction of any protein that we stain for. Again, what was interesting, I detected that the upregulation of EGFR and phospho-AKT in all the fraction of the cells on the tissue upon gefitinib treatment. But when we have BRG1 wild type and followed by gefitinib treatment, there was a reduction in the intensity or the expression of all of these markers in all the fraction of the cells on the tissue. Again, suggesting, yes, the interaction between EGFR and phospho-AKT is very important. And we think that BRG1 is the conclusion from AIM3 is, yes, BRG1 mutation actually determines the sensitivity of wild-type EGFR non-small cell lung cancer cell lines or patients. And the contribution to this resistance is through governing or controlling the interaction between EGFR and phospho-AKT. Which leads us to our final model. When we have wild-type BRG1 and treat them with EGFR TKIs, the total EGFR will be reduced. Therefore, it will not be able to, it's reduced. How can it bind to phospho-AKT to form the complex? That's why they are sensitive here. But then when we have BRG1 mutant and treat them with EGFR TKIs, the total expression of EGFR will not be reduced. It either stays or it gets upregulated, which allows EGFR to bind to phospho-AKT and form the complex and deregulate all the expression of genes that support the cancer cells to live. Therefore, to disrupt this interaction, we need to hit them with AKT inhibitors to disrupt this complex. And here the proposed treatment that we have it in this table. If we have both wild type and wild type BRG1 and EGFR, they will be sensitive to EGFR inhibitors, as well as the BRG1 inhibitors, because we used SI-BRG1 here, and the combination of both, actually, we will have better treatment outcome. But what we added, this is the the information that we added to the literature is the mutant BRG1 cells, they are not responding to these therapies. They should be treated with AKT inhibitors. And we know in the clinic that the mutant EGFR patients, they are already responding to EGFR inhibitors. But both the mutation of BRG1 and EGFR is not the case, because the mutation of BRG1 and EGFR are mutually exclusive. Here is the important key conclusion from our studies, that we know EGFR is an oncogene and it's a molecular driver of cancer initiation and progression. But there is a contradicting ideas in the literature about the role of the functional role of BRG1. For example, the tumor suppressor in breast cancer and hepatocellular carcinoma, they are known to be oncogene, but is known to be a tumor suppressor in breast cancer. But only in the different stages of pancreatic carcinoma, BRG1 in the same cancer, BRG1 can have dual function. So the question here is, does BRG1 acting like a tumor suppressor in lung cancer? According to our data, I would say BRG1 may possess both the roles. Depends on the different stage of tumor genesis. That's how tumors are taking advantage from the expression of BRG1 on and off during the tumor development and tumor formation. To further understand this role and all the other questions that this great project opened up to us, here I'm just highlighting 3 important questions. One of them could be, we need to know the role of BRG1 in the expression of the kinases and phosphatases. Because when we test, we have done all our work on EGFR, but here is the other receptor tyrosine kinases that they are known to contribute to the resistance of lung cancer and other cancers to EGFR TKIs, like HER3 and MET. And to our surprise, that the expression of HER3 and MET, they are almost similar to what we saw in the gefitinib treatment. Because when we have BRG1 wild type, the expression of these markers are reduced. And also, I would like to point out that the EGFR wild type patients now in clinic, they are treated, the primary treatment for them is chemotherapies. But the question here is, does negative relation between BRG1 and EGFR is important in chemotherapy too, or not? And further, we know that BRG1 is very important in homeostasis and mammalian development and tissue maintenance. And it has a role in EMT, epithelial mesenchymal transitioning. So here are the beautiful pictures from my spheroids that I grew, and I just want to point out one great observation, that regardless of the size of the spheroids, we can see that the core of the wild type BRG1s, the ones that they are wild type, they are kind of condensed and packed. But we don't see that pack, or they are loosely, the loose connection between the cells in the spheroid of BRG1 knockout cells has been observed. So we are really eager to see what is the role of BRG1 next in these aspects. Well, this great project, wherever I went to present my data, it allowed me, actually I won all the awards wherever I went. And it also, based on this great finding, I wrote a couple grants that I got one of them funded from the Cancer Center, and I would like to thank them for supporting my thesis for 1 year. And then I have got these publications in the past 4 years in Dr. Ramesh's lab, because I was not only working on BRG1, I was also working on a project on melanoma with the targeting RAS-RAF pathway to introduce HUR, human antigen R, and RNA binding protein as a new molecular target for melanoma therapy. And we just submitted this manuscript. And then we are preparing to, we are now preparing the manuscript for the BRG1. Here, I would like to thank each and everybody that their names are mentioned on this slide. And hopefully I haven't forgotten anyone. Although I was leading this project, but without the help and support of these people on this slide, I would have never made it here. First of all, with my lab members, all of them, the former and the current, and especially Dr. Akil, he has been really, really, really helpful, and I have learned a lot from him. Thank you so much, Akil, because you are an amazing scientist, and I can't wait to see what future holds for you. And the Department of Radiation Oncology, that I'm going to leave Dr. Munshi for now, but Daniel and Meghna, and our statistician, Dr. Zhao, and my committee members. I would love to thank all my committee members for their help, their support, to push me to critically think more. And the Department of Surgery, Dr. William Berry and James, they helped me a lot with creating the stable cell lines. And the core facility, Dr. Morley and Dr. Carmen, they helped me a lot with the quantification of the IHC slides. And Dr. Roberto Pizza and Roderico, thank you so much, guys, for saving my life and helping me with the creating the knockout cells. And the graduate college, I would like to thank deeply, deeply thank Dr. Pereira and Dr. Eyre and Dr. Eric Howard. Thank you guys so much, because at the beginning of my program, I was struggling, and they were there for me to support me, and because of them, I am here today. And the pathology department, Dr. Michael, Dr. Zhao, you are an amazing person. And our department secretaries, Rose, and our former department secretary, Milbury. Thank you guys so much. And Dr. Graham Willie from the OMRF core facility. And all the funding agencies, and also the Cancer Center, that they supported my thesis for 1 year. But last but not least, I would like to come on, I can't do it, right? First, I'm going to start with Dr. Munshi. Sorry, Dr. Ramesh. Dr. Munshi, you are an amazing scientist and a great friend. And all I can say is, during my grad school, I have been very excited about BRG1 project. And I want to say that thank you for introducing BRG1 to me. And BRG1 and I owe you a lot. And now to Dr. Ramesh. Dr. Ramesh, you are tough and very smart, but through communication and flexibility, we made such a great job here that we are proudly saying it in front of like all the family and friends around the world. I learned a lot from you, and you have been very flexible. You have been like someone like a dad. Although I maybe frustrated you, or because I was naughty, I was trying to figure everything out, and you had to like pull me back, say, Rebaz, no, come, let's do this one, and then we will think about that. So I have learned a lot from you. I can't thank you enough for what you have taught me. And I can say that you have prepared me for my entire career. And this road has been a really long road for me. It's been almost It took me almost 12 years to get here. But without all my mentors, all the way from undergrad to my master's, to my working time at the KISSR, to my Germany scholarship, and Dr. Weichin, Dr. Weichin and Dr. Howard, basically they changed my life. And thank you guys so much. And Dr. Ramesh, I just said you prepared me for my career, right? And my life mentors, my parents, I love you guys so much, and I miss you guys so much. And my siblings. Okay, I gotta do this.
We miss you too.
I wanna tell my dad that I have written here in Kurdish, so hopefully he can read it. Thank you for teaching me how to be strong and at the same time very flexible for myself and my family. To make all of us happy. And to my mom, I want to tell you thank you. Thank you so much for teaching me how to be a leader wherever I went, wherever I go, and for my stage, I can be a leader, I can lead. But when I'm a leader, you taught me how to not forget my root. So give hand to others that they need help. And also, I would like to thank all of these people. My roommates, my family, first of all, from my father's side and my mother's side, my American family. They were calling me their brown son from the other mother. And my roommates, all my friends, especially Lane. Lane, all I can say about you is our friendship needs to be written and studied in book, because you taught me a lot when I started my PhD and started my life in America. And last, I want to dedicate my thesis work to my beautiful grandmothers that I didn't get a chance to meet them before they passed away, and also my uncle. With that, I would like to thank your attention, and I will take any question. We all are here just to save life and reducing pain. Thank you so much.
Thank you, Rebaz. Thanks for that emotional at the end. First of all, I need to congratulate. Let's all give a big round of applause to Rebaz. You can do visually, or you can do the thumbs up on the chat box. So as I said in the beginning, so first let's make the committee members give them time to ask questions, and the audience, if you have any questions, just type it in the chat box so that I can ask Rebaz to answer those questions. So committee members, if you have any questions or anything to say, please, it's your time now.
So Rebaz, congrats on a great presentation. It was really fun to watch you do that. Quick question for you. You've shown in your thesis that gefitinib and these tyrosine kinase inhibitors for EGF receptor, they cause BERG1 protein and mRNA to go up.
Right.
You think that's all downstream of EGF receptor signaling? Or you know, do you think it's a direct effect of EGF receptor signaling, you know, going to the nucleus and directly stimulating BERG1?
So based on, this is a great question, because I thought about that, and the answer is not going to be straightforward. Because just looking at my data, we can see when we knock down EGFRs genetically, BRG1 protein doesn't change. But when we treat the cells with gefitinib to target EGFR, BRG1 goes up. So that means it could be the effect of the whole kinase signaling pathways that may lead to affect the expression of BRG1. That's why in the future direction, we really need to like pay attention to all the phosphatases and kinases that's regulated by BRG1.
So I guess
the answer is not direct effect.
Yeah.
It's not.
But as a quick educational follow-up for me, gefitinib is very specific for EGF receptor, right? It doesn't act on other tyrosine kinase receptors. Is that correct?
Right. That's correct.
Okay.
And I just follow up a little in this because I had the same question, and I want to know: do you think that is the TKIs affect stability of BRG1 at all? Because it looks like sometimes when you overexpress it and you treat with the TKIs, with GAF, you see an increase in BRG1 protein, and it's the recombinant protein. So that's not transcription, obviously. And so I had the same question that Courtney did.
That's a very good question, but we haven't explored whether all we could say that is when we treat the cells with gefitinib, we see upregulation of BRG1 both at the mRNA and the protein level, right? When it's wild type, but not when it's mutant. We had that observation. Maybe the mutant one is going to be stabilized by the effect of EGFR gefitinib treatment, but we haven't done that experiment.
I question, Rebaz. Congratulations for great talk. So my question is related to AKT. So how does the AKT bind to EGFR?
Do you want me to pull it up?
Yes.
I have yes. That's a good question because nobody, they have barely touched it, but they haven't actually, yes. So we did some PIMOL studies, which is not, can you see, Dr.?
No, Rebaz, you want to put on the share screen because right now I don't think we are seeing it.
Here?
No.
We don't see the slide. So you might want to go back to the share screen.
Oh, okay. Yeah. Well, to answer that question, Dr. Wu, yes. How does it bind to each other? There are, based on our PIMOL experiment, we have found a few actually candidates, the amino acid candidates that they could be. Can you see it now?
No, we're still not seeing it. So are you controlling it or is Rose controlling it?
There you go.
Yeah, there we go.
Yeah, based on our PIMOL study, which is not, I haven't finished it yet, but we can see phospho-AKT and EGFR have really, really nice docking points that they can bind to each other. And here, like I just maximized the potential amino acids that they could bind to each other. And what is interesting, the phosphorylation side of AKT are in this side of the AKT. But still, from the unphosphorylated side of AKT, EGFR is able to bind.
Okay.
But of course, this needs more studies to figure out which amino acid is actually binding, is the potential bond between the phospho-AKT and EGFR.
So there's EGFR kinase domain?
This one, yes.
So I think I use also the mutant EGFR does not bind to AKT, right?
The mutant EGFR,
yeah.
I have not done that because throughout my studies, I have been focusing on wild type EGFR.
Okay.
Yeah.
Nice talk, Rebaz. I have a quick question. So you mentioned the BRG1 mutant. I just wonder what type of mutation it is, and is the mutant still functional?
So the mutation of BRG1 has been shown to be nonsense mutation that introduces the premature stop codon that stops somewhere before or in the middle of the before bromide domain or in the middle of ATP domain, the ATPase domain. So we will have the rest of the protein still expressed. But since these two domains are very important in the function of the protein will be unfunctional when we have this mutation.
So it's a truncation.
It's a truncated protein that we have been detecting in our experiments. That's true.
Okay.
So Rebaz, to follow up to that, there was, I think it's figure 4, and I was a little concerned about that because you put that cell line together, so A549 together with the overexpressor, and it looks like BRG has the same size. How do you explain that?
Well, those are done on the same, first of all, BRG1 is 220 kilodalton. Unless you have a really nice separation, you won't see that when you overexpress put back wild type, you won't see a clear cut separation, first of all. Second, I actually have one of my data I can actually show you here, if you want me.
Yes, please.
Here. Can you see my?
Yes, I do.
Yeah. Here, because I got a nice separation, you can see that when you overexpress BRG1, the lower part is the one that's the truncated version, and the overexpression is just sitting on top of it.
I see.
But it was not common to get this kind of blot all the time just because of the separation. I had to sometimes I had to run it way down to get this separation just because BRG1 is 220 kilodalton, we wouldn't get that nice separation.
So do you know what is the size of the truncation? It's okay. I mean, this satisfies me. If it is that big, I understand that, but okay.
Okay, thank you.
So I have another question. You mentioned, by the way, and I am interrupting, so congratulations, you did great, and you're a fantastic scientist, and even a better person, judging by the number of people that was here today. So
thank you.
So my question, going back to that, you mentioned that siRNA sent to BRG1 sensitize the cells to gefednic, but then you use a knockout and actually promote resistance. So just enlighten me a little bit. So what's going on?
Okay, I'm going to share my screen again.
That shows how much he doesn't present at the back end.
What was that?
I made sure I have slide for all the questions, so.
That's good, that's good.
Right here. Can you see it?
Yes, yes.
Okay, so I actually want to based on your, maybe we can have like an open discussion about this slide, because if we take it separately, EGFR is an oncogene, right? But BRG1, there is a mixed idea about the role, the functional role of BRG1, right? But in our project, in my thesis, what we found is the negative relation between these two. That means when you have EGFR dominant, BRG1 will be less. Therefore, and this is all in the mutation is involved. So when we have EGFR more, it will dominate over BRG1, and he will be the one who is supporting survival. But when we have BRG1 wild type, sometimes it dominates over EGFR. So that's why when it dominates to provide survival, we need to target BRG1 too, because the cells are depending on BRG1 to survive in this case. And how to make this balance in the cells, that's not known between BRG1 and EGFR. But clearly our data shows that it can have both function based on whether BRG1 is mutated or not mutated. Or I have another idea actually. Maybe BRG1 role or the chromatin remodeling complexes in general, they don't fall into the tumor suppressor and oncogene roles. Since they are leaders, since they are very important in the global gene expression, they may be, based on their expression, they may be promoting other oncogene and tumor suppressors, but not themselves, if that makes sense.
Well, my problem, I guess, is that we're talking about the same cell line. So I understand that, you know, at different stages and at different situations, the same protein can work as a tumor suppressor of an oncogene, but you use the same cell line and you got pretty different results. And that, you know, I'm still a little puzzled about what's going on, if it is a dose issue or so. Yeah.
Dose issue of
BRG1.
Yes.
When you knock it out completely, but so you should follow on that somehow, because it's
I think
it was confusing to me, at least.
Right, I will definitely do that. And just to clarify what I just said is, yes, the dose of BRG1, actually we have done that in the lab. The dose of BRG1 is very important in the treatment response. And even if we take it to the expression of EGFR, let me show you another piece of data. That we created TET-inducible BRG1 A549 cells, right? So that would tell us what is happening. Do you agree with me?
Okay.
Let me find it. Yes. So in here we can see that this is the TET-inducible A549 BRG1 overexpressing cells. So when I treated the cells for 1 microgram of dogs, okay, we have this much upregulation. This is a nice separation, by the way.
Yes, that's good.
Right? Yeah.
Agree.
Yeah, so we have this much expression of BRG1, which is...
So what is the bottom band?
No, the upper band.
But what is, oh, the bottom band's the truncated one.
We asked this question and we still don't know.
I don't know.
We don't know.
Okay.
Actually, Dr. Ramesh pointed that out. But this is the overexpression of BRG1 after 48 hours that was not able to shut down yet EGFR expression. But when we waited longer, we had more expression of BRG1 and then EGFR started dropping. And if you think about this phenomenon in the treatment response of you, maybe the dose, the existence or the dose of BRG1 at different stages will have a lot of impact on gene expression, which will eventually basically define the treatment outcome.
So if so then what's going on with the 24 and 48 hours differences that you see? You didn't present that data here, but you see differences in BRG1 expression, but it's still viability, it's down at both time points. So that wouldn't go with your theory that it's a dose response.
Or maybe at the, you mean we see the upregulation only at 24 hours, not at the 48 hours?
Oh, BRG1, that's correct.
Right.
But then, like you said, the effect on viability seems to be the same or even more, right? So.
Right. So we asked this question and we still don't have an answer for, but it could be that in theory, we are thinking that maybe the first hit of BRG1 is important to change the whole gene expression profile that can put the cells in a different state and reorganize themselves, and they will go with that.
Have you done RNA-seq at all? No.
No.
Thank you.
Rebaz, can I ask another question as well? And it's a little bit similar to what Maria has been asking about, and it kind of comes down to me, it's an unexpected difference between gefitinib treatment on cell viability. So you get cell viability reduced when you treat with the TKI, but not if you knock down EGF receptor. So help me out with that. I mean, if it's kind of all about EGF receptor signaling and the effect on BERG1 levels, why is there a difference?
Can you elaborate more? I didn't actually understand the question.
Yeah, sorry. So in your thesis, you mentioned that gefitinib treatment reduces cell viability. This is in your 358 cells, the wild type cells, right?
Correct.
But you mentioned that EGF receptor knockdown does not reduce viability, at least not to the same extent, if I read that correctly. Is that true?
Like knocking down EGFR.
EGFR, yeah.
Yes, that's a very good question.
Yeah, so why aren't they behaving the same, given your final model? It seems like the whole goal is to get rid of total EGFR so that it can neither signal nor go to the nucleus and act as a transcription factor. So it seems like knocking out EGF receptor would be the best way of
right. So if we take out EGFR, that's a question that I have been always asked. Why, when we treat H358 cells with SI-EGFR, they don't respond?
Right, right.
Because they have BRG1 to support their survival. They are not depending on EGFR to survive, right?
So that's my question. Have you done a double knockdown of EGFR and BERG1 to prove that the BERG1 is what's allowing those cells to survive?
I have not done that.
Okay.
But it's the survival switch. To me, is a survival switch. If we have mutant BRG1, those cells are not depending on BRG1. Rather, they depend on EGFR. So when you take away the EGFR from them, they will be sensitive. But when we have wild type BRG1, that's dominated. They don't care if you take EGFR away from them or not. But to answer your question, we haven't done the dual.
Yeah, I think in theory, you know, that what you just suggested fits your model. But I think seeing that proof of that would be powerful to support your model.
I agree.
Okay, any other questions from the members?
Okay, just a
So,
quick question.
so it's
Yeah, Joe.
BRG1.
Dr.
Yeah,
Joe has a
me,
question.
right?
Yeah,
Okay.
Joe, we can
Great
hear you.
job. You're well prepared, Rebaz, and we are so proud of you. Just a quick question about you're dealing with here interplay of EGFR.
Dr. Zhao, I actually cannot hear you very well. I'm sorry.
Okay. I just thinking about general implication of your study in terms of other tyrosine kinase receptors. You alluded a little bit about HER2 or HER3. Can you speculate a bit more? But just not limited to EGFR here, but maybe interplay of BRG1 with other tyrosine kinases.
Exactly. That is, well, first we started with EGFR, and now we looked at the other receptor tyrosine kinases that is already known in the literature, that like here 3 and c-MET and FGFR and all of that. So we see kind of like similar, in two of them we see similar response or similar phenomenon as EGFR. So the question here is, we need to know the global effect of BRG1 on the tyrosine kinases. That will give us a better idea for treatment response or treatment option, or to develop new therapies. Or at least for now, we can screen patients or cell lines for BRG1 mutation as of now, and based on their mutation, we can say the mutant ones need to be treated with AKT inhibitors, but the wild type ones could get benefit from the EGFR inhibitors. But the global effect is very important, not only tyrosine kinases. I am thinking the phosphatases too, because I have done a couple experiments that in the knockout cells, we have downregulation of two important phosphatases that they are really important in the RAS-RAF pathway phosphorylation. I think I have the data somewhere. Here we go. So based on Western blot, I have done here 2, 3, FGF, and c-MET. But for the phosphatases, we had 2 these candidates, PTPRB and TPTE. These are the phosphatases that they are very important in the RAS-RAF pathway. So what is the global effect of BRG1 on the phosphatases that eventually they will affect the phosphorylation and dephosphorylation of the kinases? That's another question that we would like to pursue in the future.
Okay,
Rebaz,
sorry, go ahead.
So when you knock down BRG1, right?
Yes.
The EGFR total protein increase, tyrosine phosphate increase, but cell survivability decrease, right?
When I knock out BRG1.
Yeah.
Yes.
Or knock down. Yeah. So what are the survival genes that they are controlled by the BRG1? How does BRG1 control cell survival? So you knock down the cell survival decrease.
I think that the overall of the picture, the overall effect of BRG1 on proliferation is not fully understood. But speaking from the literature, BRG1 is very important in the cell cycle, for example. BRG1.
So Rebaz, the question is,
are there specific target genes?
Yeah, I think what are the genes that are known to be important cell cycle or cell survival? And is there any information in the literature, or have you evaluated any of the survival proteins like BCL2 or cyclin D or anything that's known to be regulated by BRG1 and known to play a role in survival or cell death?
Yes. So I have done a couple of markers. Yes. So this data is actually very important because it tells us that the BRG1 has an important role, not only in the expression of EGFR, but also in P27, which is these are all cell cycle regulators. So yes, BRG1 has an impact on other important cell cycle or proliferation markers for sure. But the global, we haven't done it. I think we could have done that with the RNA-seq or these kind of experiments.
Any other questions from the members? And I think it might be time to open it to the audience. I guess there's a baby asking a question with us.
Well, I have BRG1. I don't think he wants BRG1.
Is there any other question from the audience? Okay. Anybody else? Gone. It's like an auction. Gone one, gone two, gone three. Okay. If there are no questions, I would like to thank all the participants and audience. I think we're going to switch off to a private discussion with the committee members. And once again, thanks you guys for attending the whole 1-hour lecture. And once again, congratulations, Rebaz, for your awesome presentations. And Rose, can you switch off all the non-committee members, please?
I just want to say thank you so much for everybody who made it to see my presentation, because as I mentioned, it took me 12 years to get here. This day is very precious to me, and you, anybody from far or from here making it to my presentation and celebrating with me, that's a big deal to me. So thank you so much, everybody, for your support, for believing in me, and whoever just like smiled in the whole way to my face, because there were days that I was down away from my family and experiment doesn't work and all of that. So every little support helped me a lot to make it through this process. So I thank you all for everything. And I promise that I am not gonna let any of you down in the future.
Well, congratulations, Ahmad. And I really still remember the first day you showed up in my lab 12 years ago. Great job. Thank you.
Thank you so much, Dr. Weichin. Good to see you.
Wait, you didn't tell him what? That you need to keep him chained and keep it to the ground state. You never mentioned that.
Well.
Bye.
Okay, so I'm good. Great job. Bye.
Thank you.
So Rose, do we log out this meeting?
Yes.
Hold on.
There's another Zoom.
Yeah, so what will happen is now the non-committee members will be turned off, and then you can ask questions or we can have a discussion in the absence of Rebaz. I just want to know what the committee thinks.
You all leave.
Yeah, but if you have,
Join another meeting.
yeah, so you're not leaving.
Yes, there is a different invite.
There's a
That's
different
what Jerry is saying. So do we leave and join the next invite?
Yes.
Okay.
This one's over.
Okay. Yes. And I am not invited to that meeting yet. If you guys wanted me, I'm gonna be here. I can come to that meeting too.
Okay, so Rose, we're all leaving out at this time.
Yes.
Okay.
If we need to invite Rebaz to the other one, then let me know and I can add him.
Okay, I didn't realize there was another link. Okay, that's fine. Thank you.
I just sent it.
Okay, thanks.
Hello, Rebaz.
Hey, Ujwal.
Great job.
Thank you. Thank you. I appreciate, man.
Yep. I think, I didn't leave.
I am relieved right now, you know?
Yeah. Congratulations.
Thank you. I really appreciate.
I think we can leave now. So
yeah, I think I'm gonna get ready for the difficult part.
Okay. Yeah, you got this.
Yeah. Thank you. Thank you. I appreciate
you,
it, buddy.
Yeah. See you. Bye.
Guys later.
Yeah. Okay. Bye.
Dayajan. بس ساقطان بم شلون ببلات انا لە باشە من ئێستا ئەڕۆم چکە لە ژوورەکەی تر چاوەڕێمەکەن دوای تەلەفۆنتان بۆکەم باشە
ئینشاءالله
سەرکەوتوبی ڕێبازگان
دەستە ماجەکەم ئامۆژنگجان
سەرکەوتوبی بەسەر ئینشاءالله
نەتیجەکەی 2 ساعتی ترە بێ ئینشاءالله
ئینشاءالله ئینشاءالله
پیرۆزە دکتۆر ڕێباز پیرۆزە زۆر زۆرم پێ خۆش بوو دەستی خۆش بێ
تۆر گیان قوربانە بم خۆشحاڵم کە ئامادە بووی زۆر زۆرم پێ خۆش
یا ڕەبی دەست خۆش
چاوی منم
مساقوم